Awesome. Okay, so I'm studying and I hit a wall and I do what a lot of us do. I tell myself 5 more minutes of Instagram might fire me up, right? But 5 minutes later, I'm not that refreshed. I'm twitchy, I'm restless, I'm almost impatient. So here's the question. Can a tiny burst of scrolling change how risky your next decisions are? And if it can, is there a 5 minute break that might bring you back? So you've seen it. Short form scrolling is everywhere on campus. It's not just entertainment. People use it when they're tired, when they're stressed, or avoiding work. If our consumption habits change how we make decisions and whether we take risks in the moment, that matters for everyday choices, be it driving, studying, or spending. And this affects students, so basically all of us with a phone. And it matters now most than ever because this is becoming the default break between tasks. So most people assume that impulsivity is just a personality trait, right? Some people are just risky. But that might not be the whole story. There might be behavioral evidence that brief mental states such as scrolling versus self-regulation can shift risk-taking right away. And that matters because it suggests risk isn't only who you are, but also the mode that you're in. So we asked, does 5 minutes of reels versus paced breathing change immediate risk-taking on the Columbia card task in McGill undergrads? We tested students from our McGill cognitive lab class, and each person did both conditions counterbalanced. And our participants completed 2 brief inductions followed by the Columbia card task, which is a validated behavioral assessment of risk. In condition 1 was what we called our brain rot condition, composed of short form videos. And condition 2 was our paced breathing, 4 seconds inhale and 6 seconds exhale. And the task was a simple game. You decide how many cards to flip to gain points, but lost cards are hidden. So more flips means that you're taking more risks. Now, what did we see? That after reels, risk-taking went up. Our McGill undergrads flipped significantly more cards than after breathing. In other words, scrolling pushes them to keep going, to chase the reward. Again, this was only in the short term and in a small student sample, so we can't fully extrapolate. But here's another twist. Risk-taking didn't mean worse scores. Final scores were slightly higher after reels. And that's interesting because it hints that at a riskier but sometimes more rewarding style in this game. And most importantly, we found that breathing helped most when it followed reels, like a reset after overstimulation, suggesting that impulsivity can be a mode. So back to that 5 minute break. Your mental mode can flip quickly. Scrolling can push risk up, breathing can pull it down. Before an exam, a drive, or a big choice, you might not need more willpower, just a better 5 minutes. So your thumb can change your next decision, but so can your breath.
Our brain is like an orchestra, and our behavior is like a symphony. If one section plays a wrong note, we might act a little off pitch. Hi, my name is Juliette. I'm a cognitive neuroscience student at the University of Montreal, and today we're going to talk about music and the brain. Contrary to popular belief, dementia is not a monolith. It's actually a heterogeneous category of neurodegenerative diseases, and each subtype is associated with a distinct profile of cognitive and behavioral decline. In Alzheimer's disease, memory is primarily affected. In the behavioral variant of frontotemporal dementia, we see deficits of social and emotional functioning. And in the semantic variant of primary progressive aphasia, conceptual knowledge is gradually lost. Different parts of the brain degenerate. Different sections of the orchestra fall out of tune. And so my research asks a simple yet novel question. Could music cognition serve as a window into these differences? Music cognition is not a single ability. It relies on multiple cognitive systems. When we hear a song, we must first process its acoustic properties, such as pitch. We then organize the sounds into a harmonic and rhythmic structure. And then we must associate the song with semantic knowledge, such as its title or its meaning. And finally, music evokes emotions, which vary from one song to another. So in other words, music cognition recruits a distributed neural network across the brain. And so I wondered, if different dementia subtypes affect different parts of the brain, shouldn't they also affect different dimensions of music cognition? To answer this, I conducted a systematic review of 55 studies representing nearly 1,000 participants across dementia subtypes, and I organized the findings into 4 dimensions of music cognition: acoustics, structure, semantics, and emotions. And what I found was that music cognition deficits in dementia at least partially reflected the subtype-specific cognitive profiles. So in Alzheimer's disease, we saw deficits of musical semantic memory. In the behavioral variant of frontotemporal dementia, we saw deficits primarily in musical emotions. And when it comes to semantic variant primary progressive aphasia, there were impairments in both musical semantics and emotions. We're not yet at the point where music can serve as a diagnostic tool on its own, but by systematically mapping music cognition onto neurodegenerative diseases, my work provides a structured foundation to better understand how complex cognitive systems break down in the brain during dementia. And I believe that if we listen closely, music can help us hear the brain differently. Thank you.
Imagine teaching a child language by throwing them a dictionary and saying good luck. Well, that's oftentimes how we treat a lot of our AI systems. We give them huge amounts of text data in a completely random order and just let them figure it out. Well, in 2009, Yoshua Bengio and his team of researchers felt uncomfortable with this disparity and started giving AI models a structured learning trajectory, what we now refer to as curriculum learning. And much curriculum learning research now focuses on deciding how to structure these curricula. And so, in 2021, Deschamps and Bauer at Columbia University took an interesting approach, ordering the training data such that it mirrors when children would naturally learn the content. They saw interesting improvements, but there was much skepticism regarding the robustness of their results. And so, we sought to further test their findings, provide an explanation and use case for their results. To do this, we trained several embedding models. These are representational models that turn words into vector representations for downstream natural language processing tasks. For one model, we fed it words in the same order that children would naturally learn them. And for another, we shuffled the training order randomly. We then compared the intrinsic stability of the word embeddings generated. Stability refers to the consistency of word vectors across training runs. If the vectors are the same each time or similar, they're said to be stable. And so we find that when embeddings generated using a child learning curricula tend to be far more stable, but only during the early stages of training. Now, why might this be the case? Well, early learned words are also a lot more frequent. Words like go, come, mom, dad. They're also less complex syntactically and more concrete, meaning they exist as real-world objects and not just abstract ideas. So we decided to decorrelate these features to pinpoint influence. And when doing so, we built frequency, phonological complexity, and concreteness curricula and trained models the exact same way. We found the largest determiner of when children will actually learn words is their frequency, with other related factors only accounting for a small effect. Now, zooming in to the resulting embeddings, we find that they actually mirror prominent theories for when children would form these same semantic spaces. Stavers and Tenabum in 2005 suggest that children form semantic anchors that scaffold the learning environment. And we see that same scaffolding effect, that same anchoring effect happening in the embeddings created with our child learning curricula. Now, what about the utility of these models? On tasks relating to child language acquisition, we see models trained using a child learning curricula outperform baseline models by over 50%. It's not only that these models mirror prominent theories in human cognitive science, their ability to accurately capture the semantic spaces of child language acquisition poise them as providing a powerful framework to study the structural evolution of the human lexicon. We show that sometimes learning like a child isn't just charming, it's computationally smart. Thank you.
Okay, so look at this image here. Most of you probably spotted the person, the architect. Maybe you noticed the hurricane, but how many of you spotted the satellite or the mountains in the background? This is the animacy effect in action. Our brains are wired to prioritize living things, and perhaps even dynamic forces that act like living things. We notice people first, we're drawn to movement and agency, and static objects, well, they kind of just fade into the background. I'm Sam, an undergraduate researcher in the Psycholinguistics and Cognition lab at Concordia. And this observation, this instinctive prioritization is exactly what my research explores. We see the animacy effect everywhere in how we scan visual scenes, how we process sentences, and even in something as simple as reading words. In lexical decision tasks, participants decide as quickly as possible whether a string of letters is a real word, and faster response times typically mean faster access to that concept in the mind. So here's what we know. Common words are recognized faster than rare words. That makes sense. But here's what's really interesting. Even when controlling for word frequency, words referring to animate beings are still processed faster than words referring to inanimates. Our brains give animate categories VIP access right from the earliest stages of word recognition. Now here's the twist that drives my research. There's preliminary evidence that natural forces, things like hurricanes, tsunamis, and earthquakes, might be processed like animate beings. They move on their own, they're unpredictable, sometimes catastrophic, and we talk about them as if they have intentions, like saying, the storm is coming for us. So my question is, what exactly earns a concept this privileged status in the mind? Is it being literally alive? Or is it having certain dynamic, autonomous, agent-like qualities that makes something feel alive to our brains? To test this, I'm comparing reaction times for words referring to animates, inanimates, and natural forces in a lexical decision task. If natural forces pattern more like animates, it suggests that our brains aren't just tuned to life itself, but anything that behaves like a potential agent in our world. Understanding this helps us see how our minds make split-second decisions about what deserves attention first. Whether it's another person, a wild animal, or a hurricane on the horizon, our brains have already decided what matters. So when you look at this image again, and your eyes still go straight to the architect and the hurricane, well, that's your brain's animacy bias at work, quietly deciding what matters most, even before you're aware of it. Thank you.
Have you ever noticed how a bad night's sleep can make even simple things feel harder? You forget where you put your keys, you lose your sense of direction, and you struggle to recall what you studied for during an exam. Now, a cup of coffee might help you stay awake, but it doesn't actually replace sleep. Because during sleep, your brain is actively replaying these processes that happen during the day and turns them into lasting memories. And what happens when this replay system breaks down? This question is especially important for children affected by Christensen syndrome, which is a rare neurodevelopmental and neurodegenerative disease, and they often exhibit severe learning difficulties, sleep disturbances, epilepsies, autistic behaviors, and a loss of independence. And this condition is caused by a mutation in a gene that encodes for NHE6, which is a protein that helps neurons develop and communicate and keep the internal conditions inside the cell just right so that neural signals can be packaged and sent properly, kind of like a thermostat. So how this single genetic mutation leads to such pronounced cognitive deficits and sleep disturbances remains an understudied aspect of the disease, and that's the gap my research aims to address. So we know that the hippocampus is a very important structure for memory. During sleep, neurons in the hippocampus, they replay previous activities through sharp wave ripples. And these ripples act like a save button for memory, and it helps stabilize what we learn throughout the day. And it helps stabilize what we learn during the day. So what happens when this is disrupted? So in mouse models, we looked at mice with Christensen syndrome. So first, we found that they had significant deficits in spatial memory tasks. So we put them in an experiment where they had to see if a displaced object was displaced or not displaced, and they failed to recognize that. And second, we looked at their sharp wave ripples, and we found that they were disrupted during sleep. So it suggests an overall weaker coordination across memory networks. So currently, we are introducing this gene back into the hippocampus to see if it can recover brain activity, learning, and cognition overall. And similar defects in NHE6 are also seen in other neurological conditions such as epilepsy, Alzheimer's disease, and also Parkinson's. So ultimately, our research in analyzing the neuromechanisms behind Christensen syndrome not only hopes to raise awareness and inform future therapeutic interventions, but also to reveal core brain mechanisms underlying a wide range of related neural disorders. And because a rough day after a bad night's sleep for us is the everyday reality for these patients. Thank you so much for listening. Thank you.